I want you to picture a scene for a second. You are sitting in a brightly lit hospital room. Maybe you or someone you love has just been in a serious accident, or maybe it's a planned surgery.
Right, a really common scenario.
Exactly. And the doctor walks in and orders a life-saving blood transfusion. So a nurse brings in that iconic plastic bag. It's, cool to the touch, heavy, filled with dark red fluid. They hook it up to the IV and you watch the blood start flowing down the tube.
And as patients, we just expect that process to be this flawless miracle of modern medicine.
Yeah, we expect it to work perfectly every single time. But here is the fascinating and, frankly, somewhat terrifying catch. That blood might have been sitting in a refrigerator for days, sometimes even weeks.
Yeah, and that reality creates one of the most profound mysteries in everyday medicine. I mean, we tend to treat blood as this universal, shelf-stable product, almost like a gallon of milk or a bag of saline.
But it's really not.
No, not at all. The biochemical reality inside that little plastic bag is incredibly volatile.
Right. Which brings us to the mission of our deep dive today. We want to know why some bags of stored blood work perfectly, seamlessly reviving a patient, while other bags from seemingly healthy donors just, break down. Why do some fail to circulate the moment they enter a patient's body?
It's a huge question.
It is. So to figure this out, we are diving into a really groundbreaking 2026 study published in the journal Cell Genomics by Keeley and colleagues. They essentially set out to map the entire genetic architecture of the red blood cells' proteins. They wanted to understand how these tiny cells survive the extreme stress of their job, especially when they're shoved into cold storage.
It's an incredibly ambitious goal. And to really appreciate what they found, you first have to understand the paradox of the red blood cell itself. It is just an absolute biological marvel.
Okay, let's unpack this. What makes it so paradoxical?
Well, red blood cells make up a staggering 83% of all the cells in an adult human body.
Wow, 83%.
Yeah, the vast majority. And each one is packed to the brim with roughly 250 to 270 million hemoglobin molecules.
That is a lot of machinery in one tiny space.
Right. And to make room for all that oxygen-carrying machinery, the cell does something really drastic during its maturation process. It ejects its own nucleus.
It just throws it out.
Literally throws it away. It gets rid of its organelles, its DNA, its protein-making factories, everything.
Okay, hold on. Let me just pause and think about that.
Huh.
You are telling me that the cell making up the vast majority of our body is completely lacking the genetic blueprints and the manufacturing plants needed to fix itself.
Yep.
So if a part breaks, it can't just, manufacture a replacement.
Yeah.
How on earth does a cell with no DNA survive the extreme wear and tear of its daily routine?
It is a massive vulnerability, especially when you consider the core mechanism of damage they face every single day. As red blood cells travel through your body, they are doing a highly dangerous job.
Right, carrying oxygen.
Exactly. They go from the alveoli in your lungs, where oxygen pressure is extremely high, down to your peripheral tissues and capillaries, where the oxygen drops drastically. This constant loading and unloading exposes the cell to massive oxidative stress.
Because oxygen, while it's obviously life-giving, it's also highly reactive, right? It likes to pull electrons off other things.
Exactly. It's corrosive. And to carry that corrosive oxygen, red blood cells hold about 66% of your body's entire iron supply inside that hemoglobin.
Oh, wow.
So you have massive amounts of iron mixed with massive amounts of oxygen. This creates the perfect storm for something called Fenton and Hypervice chemistry.
Okay, what exactly is Fenton chemistry in this context? Let's break that down.
In plain terms, it's an iron-dependent chemical reaction that generates highly toxic free radicals. The natural autooxidation of hemoglobin generates a continuous stream of toxic superoxide. We're talking 1 to 2 micromoles per second per liter of packed red blood cells.
That sounds like a lot.
It's essentially cellular bleach being produced every single second.
So if we use an analogy here, it's kind of like using a highly reactive rusting iron bucket to carry highly volatile rocket fuel, being the oxygen, through a marathon.
That is exactly what it's like.
And you have to do this all without having any repair kits or spare parts on hand because you threw away your nucleus. That is just a terrifying job description.
It really is a hostile work environment for a cell.
But here's what I don't get. How does this damage actually manifest when the blood isn't running a marathon. I mean, when it's just sitting still in a blood bank bag in a refrigerator, it's not picking up oxygen from the lungs anymore.
And that is the really deceptive part about cold storage. When you put blood in a hypothermic environment, the cold slows the chemical reactions down, sure, but it absolutely does not stop them.
It's still rusting.
Right. Over the days and weeks it sits there, the blood experiences what transfusion specialists call a storage lesion.
A storage lesion?
Yeah.
What does that actually look like on a molecular level?
It's the progressive accumulation of biochemical and physical damage. So the lipids in the cell membrane undergo lipid peroxidation, which is, well, a fancy way of saying they essentially go rancid.
Yuck.
Yeah. And the proteins inside the cell become oxidized, warped, and damaged.
And I imagine this directly impacts how well the blood works when you finally transfuse it into a patient. How do doctors actually measure if a bag of blood has gone bad?
They look at a crucial clinical metric called post-transfusion recovery, or PTR.
PTR, okay.
Yeah, it simply measures how many of those transfused red blood cells are actually still intact and circulating in the patient's bloodstream 24 hours after the IV is hooked up.
So a severe storage lesion means a low PTR.
Exactly. The damaged cells just break apart, and the patient's immune system clears them from the body immediately. It's as if the transfusion barely even happened.
Wow. But the Kiel study found something incredibly surprising about how the cell tries to fight back against this storage lesion, didn't they? Even while sitting in the bag.
They did. Despite having no nucleus, they found these stored red blood cells have a highly active proteome.
Right, and a proteome is basically a cellular garbage disposal.
Exactly. What the researchers saw was that this degradation machinery is actually upregulated, kicked into high gear during storage. Since the cell cannot build new proteins to replace the broken ones, its only defense mechanism is to rapidly chop up and recycle the oxidized, damaged proteins before they clump together and destroy the cell from the inside out.
Okay, so let me just pause here to do a quick recap for you all listening at home before we move on. Number one, red blood cells lack a nucleus, meaning they literally cannot make new proteins. Number two, the combination of massive iron and oxygen creates daily toxic superoxide production through Fenton chemistry.
Right.
And number three, cold storage accelerates a storage lesion, which destroys the cells and lowers the clinical success rate, or PTR. So here's a question for you, the listener, to chew on. Imagine you are a cell that cannot build new parts to replace the broken ones. How would you rely on your original genetic programming to survive?
It's a great question. And to answer that, the researchers had to figure out how to look at the genetics of a cell that has no DNA.
Which sounds impossible. How did they do it?
They designed an experiment on a massive scale. They didn't just look at a standard identical lab mouse. They used 350 diversity outbred mice, often called JDO mice.
Diversity outbred meaning they aren't those identical white mice you usually picture in a lab.
Exactly. These mice descend from 8 very distinct founder strains. They capture a vast, sprawling range of genetic diversity, much like a human population. Every mouse has a unique genetic makeup.
Okay.
So the scientists took fresh red blood cells from these mice and then took other cells and stored them for 7 days to simulate human blood bank conditions.
And to see what was happening inside, they used advanced mass spectrometry. For those who might not know, this is essentially a tool that weighs and identifies individual molecules with extreme laser-like precision, right?
Right. And the sheer volume of data is staggering. They tracked over 20,000 different pieces of proteins called peptides, mapping back to about 2,350 unique proteins.
That is a massive data set.
It is. And this is where they looked for QTLs, or quantitative trait loci.
Okay, let's explain QTLs.
Sure. A QTL is simply a section of DNA that correlates with a specific trait, in this case, the abundance of a certain protein. Now, in normal tissues that have a nucleus, like your heart or kidney or liver, most protein regulation is local. We call those cis-QTLs.
Meaning a gene sits on a chromosome and acts like a local manager, directly controlling the production of its own protein right there in the neighborhood.
Exactly. But what's fascinating here is that in the red blood cell, the architecture is completely flipped. They found that trans-QTLs, distant genetic regulators located on entirely different chromosomes, are pulling the strings.
Wow.
Yeah, these distant regulators are 5 to 7 times more prevalent than the local ones.
But wait, hold on. I need to push back on this. If red blood cells don't have DNA and they don't have the ribosomes to read that DNA, how are distant genes controlling them at all?
It's brilliant, really. This flipped architecture is the ultimate signature of a cell that cannot synthesize new proteins de novo.
Okay, explain that.
Because the mature red blood cell can't make anything new, the protein levels we observe aren't being dictated by a local gene pumping out fresh RNA instructions. Instead, they are dictated by long-distance systemic interactions that happen before the cell matured, or by processes acting on it from the outside.
Ah, I see.
They're dictated by the rate at which proteins naturally degrade and by physical modifications that alter the protein while it circulates.
That makes total sense. It's kind of like a master clockmaker building a complex watch, winding it up and locking the case before sending it out into the world. The watch has no factory inside it to make new gears, but its behavior, how fast it ticks when it winds down, is still entirely dictated by the distant clockmaker's original design.
That is a perfect analogy. The genetics are acting from afar, setting the initial conditions and the degradation rules.
Did they find that male and female clockmakers, so to speak, build different watches? Like, does blood store differently based on biological sex?
They did look at sex differences. Interestingly, the most extreme chemical modifications inside the bag happened to everyone regardless of sex, but they did find differences in unmodified proteins.
Like what?
Specifically, male mice showed an enrichment in certain complement activation proteins, like the C9 protein. These are proteins that link back to systemic immune and coagulation pathways.
Meaning male and female blood might interact with a patient's immune system slightly differently after sitting in storage?
Precisely. It's a subtle difference, but it highlights just how complex the fluid in that bag really is.
Okay, time for another quick mini recap for the listener. So 1, the study used 350 genetically diverse mice to map red blood cell proteins using mass spec. 2, red blood cells are uniquely dominated by trans-QTLs, meaning distant genetic regulation. And 3, because they can't synthesize new proteins, variation is driven by degradation and systemic factors. So here is the question for our audience. If distant genes are pulling the strings of these circulating red blood cells, what specific hubs in the DNA are acting as the master puppeteers?
And the study actually mapped those out. They found 4 major cities, or genetic hotspots.
Let's go through them.
These are dense regulatory hubs on different chromosomes that orchestrate the entire red blood cell ecosystem. The first 2 hubs, located on chromosome 7 and chromosome 11, are the genes responsible for the beta and alpha chains of hemoglobin, the HBB and HBL loci.
Now, we normally just think of hemoglobin as the oxygen carrier, right? The rocket fuel transport.
Exactly. But the study showed these hemoglobin genes are present in both fresh and stored blood, and they act as massive regulatory hubs. They interact directly with structural proteins like BAN3, which is the linchpin protein holding the cell's outer membrane together.
Wow. Oh.
And they also interact with crucial antioxidant enzymes like catalase and GPX1.
So hemoglobin isn't just a passive fairy, it's actively regulating the cell's structural integrity and its defense systems.
Yes.
What about the third hub?
The third hub is a gene called MON-A, located on chromosome 9, also found in both fresh and stored blood. This one is vital for vesicular trafficking.
Vesicular trafficking.
Essentially, it controls how the cell packages up its toxic waste into tiny vesicles to expel them. It also dictates macrophage iron recycling, meaning it controls how the spleen recognizes and consumes old, damaged red blood cells to safely recycle their iron through extravascular hemolysis.
Okay, but here's where it gets really interesting, the fourth hub, because this is where the mystery of the blood bank bag really gets cracked open, doesn't it?
It absolutely does. The fourth hub is a gene called STEAP3 on chromosome 1. And the fascinating thing is, this genetic hotspot only appears when you look at the data for the stored red blood cells.
Wait, it's completely invisible in fresh blood?
Completely invisible. STEAP3 is a storage specialist. The protein it codes for is a ferroductase, an enzyme that reduces iron, changing its chemical state.
Okay.
When the blood is placed in the cold, stressful environment of a storage bag, genetic variation in this single STEAP3 gene suddenly dictates everything.
Like what?
It controls the iron-dependent oxidative stress. It triggers the release of those waste vesicles using APO proteins like APOA1 and APOE, and ultimately it correlates strongly with the post-transfusion recovery rate we talked about earlier.
So it is quite literally a hidden switch that determines how well your donated blood will survive outside your body.
Yes, and what connects all 4 of these hubs, the hemoglobins, the MON-A spleen interaction, and this hidden STEAP3 iron switch, is that the proteins they regulate are heavily enriched for proteome components.
Circling back to the garbage disposal.
Exactly. It proves that the degradation of damaged proteins is the central pillar of survival for a red blood cell in a bag.
All right, recap time. 1. 4 genetic hubs control these cells: hemoglobin alpha, hemoglobin beta, MON1A, and STEAP3. 2. MON1A controls how the body clears out damaged cells. And 3. STEAP3 is a hidden switch that only activates during storage, regulating iron toxicity and cell survival. So, listener, knowing that hemoglobin itself is acting as a regulatory hub, how exactly does a protein meant to carry oxygen defend the cell against the very toxic stress it creates?
The answer to that lies in something called post-translational modifications, or PTMs.
Okay, let's break that down. Post-translational modifications. If the original protein is a newly manufactured car rolling off the assembly line, the PTMs are everything that happens to it out on the road, right?
That's a great way to visualize it.
It's the dents from a fender bender, the rust on the undercarriage, maybe an aftermarket upgrade or two.
Exactly. PTMs are chemical changes that alter the protein after it's already built. The study tracked 20 different types of these chemical changes, and they found that oxidation and deamidation were the most abundant by far.
Which makes sense given all the oxygen and iron we talked about. Molecular rusting.
Right, and this reveals the hidden secondary function of hemoglobin. Because there are hundreds of millions of hemoglobin alpha and beta chains in every single cell, they act as massive PTM hubs. They function as a giant biochemical sink, or a sponge.
A sponge for damage.
Precisely. They deliberately absorb the oxidative hits. They take the dents and the rust in order to protect the much less abundant but absolutely critical metabolic enzymes from being destroyed.
So the cell essentially sacrifices its own hemoglobin to keep its core machinery running.
Yes, and the researchers found a fascinating genetic quirk in some of the mouse populations that proves this sponge theory perfectly.
Oh, the cysteine variant.
Yeah, there is a variant in the HBB locus, the hemoglobin beta gene, that introduces one extra cysteine amino acid at position 13.
Cysteine. That's a specific building block of proteins. What makes it special?
Cysteine is highly unique because it has a reactive sulfur group attached to it, known as a thiol. Think of it like a highly reactive chemical magnet.
Okay, so adding this extra sulfur magnet changes the chemistry of the sponge.
Dramatically. The researchers saw that this extra cysteine acts as a direct sink for glutathione.
And glutathione is the cell's master antioxidant, right? It's basically the cellular fire extinguisher.
Exactly. The mice with this specific extra cysteine variant had altered levels of glutathione disulfide. The underlying genetic architecture of their hemoglobin directly manipulated the cell's entire antioxidant balance.
Wow. Okay, quick recap. 1, PTMs are chemical changes like oxidation that happen to proteins after they are made. 2, hemoglobin acts as a massive sponge to absorb that oxidative damage. And 3, a specific genetic variant adding an extra cysteine alters the cell's entire antioxidant balance. So listener, if adding an extra cysteine changes the whole antioxidant balance, what happens if we remove the most important cysteine from the equation entirely?
To answer that, the scientists executed what I think is the ultimate proof of concept in this paper. They genetically engineered humanized mice.
Humanized mice, meaning they went in and completely replaced the mouse hemoglobin genes with human hemoglobin genes.
Yes. They are running human blood mechanisms inside a mouse body.
That is wild.
It is. And they created 2 distinct test groups for this case study. Group 1 got the normal, healthy human hemoglobin.
Okay.
This normal version contains a highly conserved, absolutely critical cysteine at position 93 on the beta chain. Let's call it beta C93.
Got it. And group 2?
Group 2 got a mutant version. In their hemoglobin, the researchers surgically replaced C93 with a completely different inactive amino acid called alanine, the C93A mutation.
So they stripped the blood of this specific sulfur magnet. Why is C93 so important to begin with? What's the why behind it?
Under normal, healthy conditions, C93 buffers intracellular glutathione. It helps maintain redox homeostasis. But under massive oxidative stress, like sitting in a cold blood bank bag for a week, C93 undergoes a violent, irreversible chemical reaction called beta elimination.
Beta elimination. That sounds destructive. What actually happens to the molecule?
It physically loses its sulfur atom and becomes a molecule called dehydroalanine.
Oh.
Yeah, without that sulfur, it can no longer buffer damage, and it can no longer help recycle other antioxidant defenders like PRDX2 dimers. It essentially throws itself onto the grenade to absorb the blow, but loses its weapon in the process.
So what was the clinical outcome for those mutant mice? The ones completely lacking that cysteine shield, did their blood survive the storage bag?
It was total biochemical chaos. Lacking that single cysteine residue caused the red blood cells to fail on almost every single level.
How bad was it?
The C93A mutant mice had drastically lower levels of reduced glutathione, or GSH. They accumulated toxic byproducts like 5-oxyproline. They suffered massive purine oxidation.
Purine oxidation.
Basically, the building blocks of energy in the cell were destroyed, which led to a huge buildup of urate. Cellular waste was just piling up uncontrollably.
So I assume their proteome, their garbage disposal, just couldn't keep up with that level of wreckage.
Exactly. Their cells showed a compensatory upregulation of the garbage disposal machinery, desperately trying to clear out the wreckage. Ultimately, both the normal humanized models and the mutant models had poor post-transfusion recovery rates, but the C93A mutant mice specifically and dramatically lost their chemical balance. Their internal redox ecosystem was entirely shattered.
So what does this all mean? We can synthesize this entire mystery down into one extraordinary mechanism. A single amino acid Cis93 sitting on a hemoglobin molecule interacts with that hidden iron-regulating gene, Stephen3. And together, they form a master redox metabolic circuit. And it is this specific microscopic circuit that dictates whether a stored bag of blood will live to save a patient or die in the refrigerator. Let's do one last recap. 1. Scientists swapped mouse hemoglobin for human hemoglobin to test cysteine 93. 2. Cis93 acts as a crucial molecular shield for the antioxidant glutathione. And 3. Removing Cis93 caused a total failure in the cell's ability to handle storage stress. So listener, how might this incredibly specific genetic knowledge change the future of medicine?
If we connect this to the bigger picture, the clinical applications are profound. This isn't just an elegant mouse experiment to be filed away in some journal.
Right, it has real-world impact.
Huge impact. Understanding the genetic interplay between STEAP3 and hemoglobin Cis93 could completely revolutionize transfusion medicine. Right now, blood banking is mostly a logistical challenge, supply and demand. But with this data, by screening donor genetics, blood banks could predict exactly whose blood stores brilliantly and whose breaks down quickly.
That's incredible. We could route the highly resilient blood to long-term storage or rural hospitals and use the fragile blood immediately in fast-paced trauma centers.
Precisely. Furthermore, this mechanism finally helps explain the wide variance we see in human diseases like sickle cell disease and beta thalassemia.
How so?
In those diseases, mutant hemoglobin variants trigger massive oxidative stress inside the patient's own body. By understanding how the normal cell uses the proteome and the hemoglobin sponge to cleave that stress, we open up completely new avenues for therapies to help those patients manage their hemolytic propensity, how likely their cells are to burst.
It really changes everything we thought we knew about a cell we completely take for granted. We've explored the paradox of the red blood cell. A cell with no DNA, navigating a river of toxic oxidative stress. And we've seen that it survives through an incredibly elegant, long-distance network of genetic predispositions. It utilizes hemoglobin not just as a ferry for oxygen, but as an active sacrificial biochemical sponge, guided by hidden genetic switches like STEAP3 that only wake up when the cell is in danger.
It's a testament to the fact that even the most abundant, seemingly simple cells in our body possess layers of complex regulation that we are only just beginning to decode.
Absolutely. So I want to leave you, the listener, with a final provocative thought to mull over. We spent a century treating all blood donations as basically identical commodities. Just match the A, B, O blood type, make sure it's safely screened and put in the fridge. But knowing what we know now, knowing that deep genetic quirks down to a single sulfur atom on a single amino acid dictate how well a blood bag ages, could we soon see a world where your donated blood isn't just matched by type, but is genetically tailor-made and specifically prescribed to a patient based on its individual oxidative resilience? The bag of blood that saves your life might be chosen for you based on a microscopic genetic clock wound up weeks before it ever left the donor's arm. Until next time, keep exploring.